Introduced to you today are two presenters. April Pike is the Interim Dean and Associate Dean of Research at the Faculty of Nursing at Memorial University. She holds an undergraduate and master's degree in nursing and a PhD in medicine from Community Health and Humanities at Memorial University. Her research expertise is in the field of genetics, hearing loss, cardiovascular disease, and qualitative research methods. Dr. Pike has a strong record of public engagement, community service, and education. She is the recipient of the Memorial University President's Award for Exceptional Community Service in 2015, as well as the President's Award for Distinguished Teaching. She is a member of the Pan-Canadian Steering Committee for Nurse Researchers, known as Canadian Nursing and Genomics. Also is Rebecca Puttister. She's a PhD student at Memorial University of Newfoundland Faculty of Nursing, and she's supervised by Dr. Joy Madigan. She holds an undergraduate and master's degrees in nursing from Memorial University. And she continues to work part-time as a direct care RN and has evolved as a research assistant with the Canadian Nursing and Genomics Group. Her master's practicum was a policy proposal for nurse-led care for individuals affected by hereditary breast and ovarian cancer. She has completing training in cancer genomic risk assessment through the City of Hope Medical Center in the United States, and her PhD project will focus on preparing oncology nurses to deliver patient-centered care and prevention informed by genetics and genomics.
Myself and April are very excited to talk to you about something that we're very passionate about, and that is engaging nurses with genetics and genomics in their practice. So just a brief overview of what we're going to talk about today. We're going to start by going back to high school biology for a minute. We're going to talk about what all this really means to healthcare and contextualize it within our own province. We're going to talk about some of the work of a group that April and I have been involved in for about a year, which is the Canadian Nursing and Genomics Group. And we're going to talk about some of our future plans and also, of course, highlight for how you as the attendees, how there's a role for you guys to become involved as well. So like I said, we're going straight back to, I don't know, biology 3201 or whatever it is. And I just like to highlight this because just to remind us that deoxyribonucleic acid, or DNA, is that molecule that carries our genetic instructions. So that codes for our particular proteins. So those four chemicals, you'll often heard them referred to as nucleotides. It's made of those A, T, and the C, and the G. And there is a reason I'm saying this today, and as we go forward, I hope, but just keep in mind, A, T, and the C, and the G. Because, and I just should say that the order of these chemicals is important because the order in which they appear is going to code for a different protein, which of course has implications for our health. So again, at the very basics, genes, we have 23,000 genes. They're those basic functional units of hereditary. So in other words, they make the instructions for exactly what we're made of, like I said, written in that DNA chemical code. So you might hear somebody say, oh, I have the BRCA gene, but it's not a matter of if somebody has a cancer risk that they have a BRCA gene. We all have the BRCA gene, but some of us inherit copies of the gene that have what we liken to a spelling error in that inherited copy. And therefore, for example, BRCA, which you've probably heard of because of Angelina Jolie, it doesn't make its job is to code for a tumor suppressor protein, which is the job is to regulate cell division and prevent cells from growing uncontrollably. But because somebody inherits a copy that doesn't make that protein the way it should, this is why they're at elevated risk of breast and ovarian cancer. And there's a lot of other different single gene variants, which of course we're all very familiar with hearing about variants now, and genetic variants from the COVID-19 pandemic. So if you're not familiar with what the word genomics means, please do not feel, don't feel intimidated or anything, because really before I started getting interested in this area of research, I really didn't understand what the word meant either. So of course, genetics is going to be very familiar to most people. We have the study of inheritance and how inheritance or a change or a variant in a gene can result in different implications for our health and disease. Genomics, on the other hand, is encompassing, now we can look at a multitude of genes and their involvement in health and disease. And also we can look at the gene interactions between other genes and how genes interact with the environment. And you'll have to excuse me because a lot of my examples are going to be talking about cancer, but even at the tumor level, we can look at the DNA of a tumor for acquired genetic mutations. And this is really transforming precision oncology the way we know it, because we can direct treatments that reduce side effects and also have longer survival risks because they're based on an individual's unique genetic makeup and the genetic makeup of those tumors. So yeah, so that's years ago when you were talking about if somebody had a lung cancer, they'd have small cell, non-small cell, and that's how you do it. But really, we know that this is a lot more complex. And really what they're hypothesizing is that there's actually a whole bunch of rare cancers versus, so rare is not that uncommon. So this slide actually, when April was presenting about X number of years ago when she was doing her PhD project, this was a slide she used. And it's really important to talk about the Human Genome Project because this was really, you know, a really big monumental thing in what advanced genomics. So it took 13 years, starting in the year 1990 to 2003 when it was completed. They actually mapped the entire human genome. So that gave us, it's equivalent to 3.2 billion letters of that A, T, and the C, and the G. So theoretically now we understand, I guess, in a quote unquote healthy person, what the intended functions of those genes should be. But of course, our understanding of this is certainly going to expand as more data is collected. And this is why data sharing is so important and international data sharing. So of course, because that it's a lot less expensive now to sequence. We have understanding, particularly in the areas of cancer and rare diseases. This is where we're really seeing the diagnostic utility of this the highest. And also in pharmacogenomics, which is when we are actually targeting drugs based on an individual's unique genetic makeup. So that results in less adverse effects and more therapeutic effects. So don't get too overwhelmed by this particular picture, but I just want to show you sort of what the process is when somebody is sent for genetic testing. Now, bear in mind, I am not a geneticist, so this is going to be a very basic explanation, but just understand the tissue, whether it be tissue or blood sample, it's being prepared, copies are being made, and you can see here, this machine is a sequencer machine. And you'll notice this gentleman here in his computer, and I'm just going to advance to my next slide. That's what the sequencing is doing. So if you're getting a multi-gene panel or your genome sequence, which would be the equivalent of a 75 million page document, that's how big our human genome is, this is what the printout would be. So we're looking at any variation in those letters as to what would be expected. So I just want to say here that despite the fact that I've shown you a very complex technology, I like to just use clinical examples with what is known to us. So for example, whether you work in acute care, primary care, we know that if we're sending a patient, for example, they're getting a CBC or an LBC, we know when we get a result by that when we need to act, or we would know why a particular sample might be being involved. But in terms of what happens in the lab between now and then, between when we send it down and it gets reported, that's outside of our scope of practice. Same as nobody would necessarily expect a nurse to be able to, or a general practitioner for that matter, to be able to interpret the nuances of, let's say, a CT scan. But we are used to working with different forms of clinical knowledge, and people would expect nurses to explain why a particular sample was being involved. And certainly we find ourselves as nurses, I'm sure many of us here, if you're still in practice, you'll recount times when a patient will come to you and say, can you explain to me what he said? I don't understand it. So we know that there's a very important role for nurses here in terms of continuity of care, and which is exactly why we're here trying to get our message out. So this is an in vitae lab report. So this is for an example of somebody who would have been sent to see if they have a cancer genetic mutation. And a lot of these are actually sent out of province to this in vitae company, which you would be familiar with if anybody's the process for prenatal genetic screening. A lot of times that is done through in vitae. So a lot of times through our healthcare, they sort of outsource it to these commercial labs. And I won't belabor this, but just because I think it's important, and even in Canada, we have some nurses who are doing some very exciting epigenomic research. So I was telling you about these A, T, and the C, and the G, but what epigenomics is actually, when you'll see these little M's, these are the methyl caps. And when these caps are methylated, they can either accelerate gene expression or turn it off. So that has a lot of effects. And I meant Dr. Nicole Letourneau, who we'll mention in a moment, she's doing studies about how different methylation and histone groups that attach to these genes, how that can be involved in immune responses, stress, and other symptoms. And actually, they've actually done some studies in Holocaust survivors showing that this methylation from trauma can actually be inherited. So when you're talking about nursing and the social determinants of health, which we all know is very relevant to our practice, we know that this is an area where nurses can be doing a lot more research. And there's exciting progress, particularly in the States, that's being done with this work. So Newfoundland and Labrador, it's well known that because we have these large families where you could see patterns of disease inheritance, we had this founder effect where we were a geographically isolated province. It's well known within the scientific community that Newfoundland is really considered a gold mine for studying genetic and genomic diseases. And these are just a few examples. This is Dr. Rahman has written about this back as far as 2003, but even in the 70s and 80s, people were talking about and using Newfoundland as a prototype. And of course, ARVC, which again, you'll see here, April did a lot of work in that in her doctoral dissertation, but even things such as we have a lot of Lynch syndrome and this RAD51C variant, which there's very strong evidence that this predisposes our population to breast and ovarian cancer. So we know that these advances in genetic genomics, we really have an opportunity to upskill healthcare providers, but also have clinical translation so that people can benefit from these scientific advancements in our province. And like I said, cancer is multifactorial, and there's still a lot about it that we don't understand. But when you consider that we do have this founder population, we have the highest cancer incidence rates of any province in Canada and highest rates of colorectal cancer, we definitely need to do a better job at identifying people who are at increased genetic risk so they can avail of screening and prevention, that there's strong tier 1 evidence that this does prevent cancer. As well, we know that there's some deafness, heart disease, ARVC, like I mentioned, some types of diabetes and psoriasis, as well as multiple endocrine neoplasia. So we're kind of a melting pot of genetic conditions here. So, and I can't take credit for this particular statement because this comes from our colleagues in the UK, but we're really moving. And like I said, the CIHR, which is the Canadian Institute of Health Research, they anticipate that within the next 10 years, everybody is going to have their entire genome sequenced and used to inform the care that they received. We don't really seem ready for that. So this is a lot of, why April and I are interested in the work we're doing today. And I just add this is a news excerpt, I believe, from 2021, when the Center for Translational Genomics, which is a partnership between Memorial University and Eastern Health, this is when the Center for Translational Genomics went live. And this is in its infancy, but the idea is that this will be a hub where we can get genomic sequencing, have it done here at home, and use that, use our founder population to yield diagnostic value and improve health, which of course we know from our health outcomes is something that's definitely needed. So right now I'm going to turn it over to Dr. Pike, and I'll be back.
Thank you very much, Rebecca. I'm just going to continue on, and I'm very happy to be here today. I've been doing research in genomics back in the early 2000s, and it sort of faded for a bit, and then it's come back to attrition now, so that looks at the significance of it. But I just want to give you a brief overview of what's been known about nursing and genomics. And there is an overall consensus in the literature that in order to provide safe, ethical, and competent, compassionate care, nurses really need to be grounded in genomics and particularly what it means for the client, their family, and their health care. So there is little Canadian research in the area of genomics. However, what has been known is that nurses do have low levels of genomic knowledge. very little formal education, and that translates into nurses not having high levels of confidence in order to perform with a genomic lens. And interesting is that in Bortkoff in 2005 actually stated that nurses need to have the integration of genomics into their care. And this continued on in with my work, but there was little attention given to the role that nurses play in genomics. Even though that, low levels of genomic literacy is a worldwide challenge, there are countries like the US and the UK that are making strides towards integrating genomics into practice that we can actually and have looked to help us support this within the Canadian context. So many of the conversations that we've been having with nurses, they've been saying, like, what relevance does this have to my practice? So I just want to give you a little bit of the flavor of the types of questions that nurses are being asked by patients. And I ask you to reflect on your own practice and think about, have I been approached in the same manner, or have I actually had some of these thoughts in my own practice? And everybody knows by the 23andMe, the genomic test report that's available. So, I mean, I've had people come up to me and say, can you have a look at this report? And can you please help explain some of the results and how that informs my decision making? Other people are really concerned about, if they've been diagnosed with a genetic condition, about the impact that it will have on their family. And this is a quite common one. Are my children at risk of getting the same cancer as me? I have my genetic test results. What should I do about my screen and lifestyle that will actually decrease my risk? Because if you have a genetic predisposition for a condition, it doesn't mean that you will develop it. It can be based on modifiable factors like exercise, diet. So there are different, how can I say it, different modifiable factors that would actually affect gene expression. If I agree to genetic testing, can the results be used against me in somehow? So people often bring up about house insurance, medical insurance, and how knowing this, and about job opportunities. For example, if you have the gene that I studied for ARVC, which causes sudden cardiac death, can I be a pilot? Now, most people don't want to be on a plane with someone who had that genetic condition, but you can understand if that was someone's lifelong dream. So it actually has huge implications, not only for someone from a financial perspective, but also from a job and career perspective, and from a longevity, because genomics is not something that happens to an individual. It happens to families, and for long after you're dead and gone. So it's cross-generational. The doctor told me I have a genetic variant. I don't understand what she said. Can you explain it? What is a polygenic risk score? I'm now in the palliative phase of cancer treatment. How can I get genetic testing for my family? So I'm sure, people are nodding there saying, okay, that makes sense. I've been asked some of these questions, but I really never thought about it practicing from a genomic lens. So based upon the research and our experiences in the clinical practice, in 2020, the Canadian Nursing and Genomics Steering Group was formed, and I'll get into a little bit about that later. But the overall goal was to help accelerate the integration of genomic knowledge into our practice. So the key factors were like to increase awareness about genomics, particularly the relevancy of it to nurses, to create the infrastructure to support genomic practice, increase research in Canada. As I said, there's very little research done in Canada, to increase our collaboration with the bench scientists and increase patient engagement and clinical innovation, which is pathways to accessing genomic information and how that process evolves and what steps are needed along the way. So in order to do this, the first step was to create an engagement framework, but actually to map out our plan to accelerate the integration of genomics into nursing practice. So it's more like a blueprint. So on the steering committee, we have the founders or the co-lead. We have Jacqueline Lemoyne. She's associate professor at Athabasca University, and she's the chair of Ontario Cancer Research Ethics Board. And we have Lindsay Carlson, who actually completed her PhD a couple of weeks ago, who's a clinical nurse specialist at Princess Margaret. We have Sarah Duo, is assistant prof at the University of BC. Nicole Latour, who Rebecca alluded before, is actually a professor at University of Calgary. Myself. And we have Anne Miner, who's advisor at Ontario Genomics. And of course, we have Rebecca Puddester, who is my PhD student and a research assistant for the CNG, the Canadian Nurses in Genomics group. So as I said previously, the purpose of the engagement framework was to create a theory and evidence-based framework, which was actually generated by nurses and will be used by nurses and other partners to promote sustainable initiatives for genomic literacy. So basically a roadmap of what to do, what are our priorities, and how to do it. And as with all good research, our research study has a strong theoretical foundation to the framework. We use Rogers' theories of diffusion and innovation, and of course, the knowledge to action cycle by Graham. And I won't get into that now, but we can certainly talk about that afterwards. So this is just a broad sweeping slide. Actually, it's our poster that talks about the engagement framework, about the aims of it, the rationale. And then I want to focus your attention to the bottom left hand about building the engagement framework. And actually, these are the 4 steps we're going to talk about that led to the identification of the 6 key priorities on the right hand side. So the first step of the engagement framework is conducting a literature review. Then we engage nurses in key stakeholder conversations across all domains of practice. We did an environmental scan to look at the gaps and the opportunities for integrating genomics into nursing practice. And then we actually consulted with our international partners. So each of those 4 things is what we'll present next. So the first part was the engagement framework review. And so here are some of the key findings that we looked to, and we found that there are key adopters and opinion leaders in genomics that we need to tap into to actually increase the relevance and uptake of genomics into our practice, particularly the oncology nurses. And this was no big revelation to us because the oncology nurses are the ones that are traditionally dealing with the big cancers. So they are the people that if you were going to do something, you would actually start there because they got the buy-in, and then you would move forward to other domains of practice. There was an assign tool that we discovered by Emma Tompkins that looked at 6 critical success factors that you can use to evaluate the success of genomics into practice. And they included enhanced education, effective nursing practice, infrastructure, professional collaboration, family community focused care, and then policies. So you can see once we understood from the literature what are the key priorities, who should we target, and what has been done to evaluate, we can start trying to formulate how we would operationalize and move forward. So just next slide. So in the literature review, we decided that engaging in dialogue with nurses can actually help us pinpoint some of our priorities and some of our challenges. We can assisting nurses and faculty to understand the importance of genomic literacy and genomics is really key, because once we can get people to really buy into the relevancy of genomics, we can motivate them to learn and then start to make room for genomics into your practice and into nursing curricula. And then there was a roadmap that actually was mirrored with the assign tool, those 6 critical factors that we could use to accelerate the integration of genomics. So basically, the roadmap is very similar to our engagement roadmap that we contextualize for the Canadian context. And very importantly is that ongoing education, all levels from entry to practice, is really critical to uptaking the genomics into practice. So not only do we need to deliver it within our nursing curricula, we also have to provide opportunities to augment that knowledge and actually bring it to life as standards to practice. So the second part of the engagement framework, we actually held virtual focus groups with key stakeholders across the country with 27 nurses. And you can see in the right-hand side, we had people from all domains of practice. We had people from administration, research, education, policy, and point of care. So what we did is that we asked them and invited them to have discussion about what are some of the clinical questions that you're being asked? What are some of the knowledge needs? What are some of the missed opportunities with interprofessional and research practice? What infrastructure required for you to consider integrating your genomics into your practice? And where do we need a network? Where do we need to start to building relationships and partnerships to make this come to fruition? The third thing that we did was we developed a policy document. So we did an environmental scan of document analysis of all professional documents across Canada. So we actually looked at 37 documents, and overall within Canada, we found there's a lack of depth and breadth of Canadian nursing documents that include the context related to genomics. And this is really earth-shattering when you look at leading countries such as the US and the UK who are developing policy procedures and integrating nursing into the curriculum. So as we were doing this, we were looking for opportunities to enhance the guidance available to Canadian nurses for the application of genomics. We actually looked at nursing professional associations such as Canadian Association of Schools of Nursing, the regulatory bodies, and we looked at special interest group bodies. We identified that the early adopter, when we looked at these specialty groups, it confirmed that the early adopter of nurses, such as oncology nurses and perinatal nurses, present the best opportunities for buy-in for research around genetics and genomics. And as Rebecca said, given our physical plant or our genetic plant within the Newfoundland, it's no surprising that the oncology nurses would be our early adopters here and our best buy-in. So this actually has been accepted to Canadian Journal of Nursing Research, and Rebecca is the primary author on that. So kudos to Rebecca. So the fourth part of developing this engagement strategy, or this roadmap to actually identify our priorities, where we should go and with who, we actually consulted with our international experts. And I'm not going to go into the slide, but these are just an example of some of the big players in nursing and genomics that we actually took our engagement framework to and said, is there anything that you could see we can add? Anything that should change? Is there anything that we should consider? So based on that, those consultations, we created our engagement framework, and we identified 6 key priorities for action. If you remember back in the poster that I said on the right lower F quadrant, these are the same ones. So the first thing was to engage nurses to see the clinical relevance of genomics that will support buy-in. The second thing was to identify and clarify unique and overlapping roles in genomics that will help us improve population health outcomes, enhance patients' experience, increase nurses' satisfaction, and see what was the best bang for our buck. And we want to help develop the nursing workforce through education and to learn more about genomic literacy, to increase and revise new clinical care pathways that would include genomics from entry into the healthcare system and to exit. One example is nurses doing a genomic health history. We need to develop the infrastructure to support professional practice in genomics, practice standards, curriculum, policy, procedures, and collaboration. And we actually have a grant gone in to look at how to develop these, the infrastructure, and look at policies, and then advanced research. So all of these priorities were identified. So our next part of our goal is to take these and operationalize them into practice. So if you just want to read more about the CNG framework, we have an article that's in the nursing research that you can actually look and read more about the good work that we're doing. And that's an open access article, so it costs nothing. So now I'm going to turn it over to Rebecca to talk more about the execution of the framework and how we're planning on operationalizing it.
Oh. All right. Yeah, so that was the labor of love for the team after probably a year, and then it's still going on after all is said and done. But like April said, now is time for the execution of the framework. And this is not just something for us as the CNG, but this is you know, all nurses in all the domains of practice. So, you know, I got thinking myself, okay, time to put my money where my mouth is. So first, I'm just going to share a little example. Because I think, as Dr. Rahman mentioned, I still practice on an acute care medicine unit at St. Clair's. So you'll recall that the first priority, we said engage nurses to see the clinical relevance of genomics. And also going back to our theoretical framework, when you talk about knowledge and persuasion, we still really have a bit of work to do to get that buy-in. So thankfully, with the support of my manager, Jackie Brockerville, and the clinical educator, Bernice Woodford, they allowed me, as part of our annual education day, to give a 30-minute presentation with the two sides of the schedule. And actually, that should be 2022, excuse me. And it was just a little slide, nursing and genomics, why should I care? So this was a very, you know, high-level overview. But again, thinking about the clinical relevance and talking about examples. So, you know, I talked about the BRCA and Angelina Jolie. And actually, I don't know if anybody's seen, but Liam Hemsworth, actually, he recently came out and said that he found out that he had a gene that predisposed him to early onset Alzheimer's disease. So some of the genetic counselors, some of the dialogue between theirs, they expect that this will be the new Angelina Jolie effect, because after Angelina Jolie went public with her BRCA, that she carried that variant, genetic counselors, there was a huge increase in demand for genetic counseling. Anyway, back to went a little off topic there, but so I used that example, but then I also framed it in terms of us not only as health professionals, but also as users of the health system as well. And I went to the app for our insurance through Eastern Health, which is Canada Life, and I had a screenshot that says, and our insurance will actually reimburse some of us for pharmacogenomic testing. So if somebody was taking, let's say, an antidepressant or some sort of other medications, there's actually a process through our insurance to have the pharmacogenetic testing, and it gives you a printout of what drugs are most likely to yield the most therapeutic effect for you. So and of course, we always have to talk about the COVID-19 pandemic, because that is the end. As you know, we've heard variants more than we even want to, more than we even want to talk about now. But that's what it is. We're talking about a variation in, you know, it's a bit different with RNA, with the genetics, with the viruses, but it's the same concept. So just to sort of conceptualize that this is, and I often go back to one of the researchers on our team, who is Dr. Sarah Duell. One of her sort of discussion papers, which is published in the Canadian Journal of Nursing Research, is called about seeing the familiar in the foreign. Because while genomics as a knowledge form is sort of unfamiliar to us, because it, like I said, from the document analysis, we know that this has not been part of our foundational knowledge really at a, you know, substantial, meaningful level. Whether it was covered in anatomy, physiology, we don't know how to apply it in our practice. So, like I said, I gave the presentations. I don't know if, you know, they're my coworkers, so I'm not sure if they were just, you know, being kind, but I think, you know, the feedback was good. You know, this was really you know, this was just whetting their appetite for it. So we know that they really didn't have much introduction to this beforehand. And I did a little tiny anonymous survey, and they, you know, they said most of it had not heard of it beforehand. And those who did, it was because, you know, I probably talked the face off them about it before. So, but these are small steps, like I said, getting at that buy-in. And hospital managers and leaders, they have an important role in that. And again, we know that, again, linking that back to the diffusion of innovation theory, it's the social systems that influence whether or not this is adopted as well. And having a gatekeeper or a champion of genomics is useful. The studies out of the UK with Dr. Kathy Calzone really confirm this. They've integrated this with people in different practice areas, and they had little champions. Not that I'm calling myself a champion, but I can certainly talk the face off anybody about genomics. And lastly, make it interesting. So I had my little genome sequence here, and again, I tried to, you know, come up with the examples that really got that buy-in. So that's just a very grassroots example of my own practice unit. So, you know, just thinking about, you know, a lot of people are probably going through doing master's projects, things like that. So even not in the context of genomics, I'd encourage people to think about how they can, you know, upskill in their own practice unit from their own burning clinical questions. So we mentioned about the GGMPS, and really not just, our examples, but, Dr. Lindsay Carlson, who's involved in a really interesting project as well out of Princess Margaret about genomic. It's actually about clinical trials and the idea of a nurse navigator having that genomic literacy for people who are in phase 1 trials, so advanced metastatic disease. But anyway, this is the big project that's we've been trying to get it on the go now for quite some time. We've encountered some delays, but it's ready to launch basically now. And then it's going to be a national survey. So when I get the full approval, there will be anybody who signed up to be contacted for research from the CRNL. They will receive an email of this survey coming through your emails from the CRNL. So basically, we know we only have these 4 studies that have been done since 2005 that show that we have that limited genomic competency. So, we're at that tipping point now. We need that benchmark across Canada, because most of these studies have been done in single center, out of single centers. So the plan, and this is a project led by April and Dr. Jacqueline Limoges as primary investigators, we're going to launch this genetics and genomics practice survey. So this is based on a validated tool, which was developed in the US. It shows good validity. There's been a lot of testing done on it. So this will come, a link or a QR code to participate in this survey will come to your email. And we want to create that benchmark. So there are 12 items on this that gives you a knowledge score. We're going to return that knowledge score to the participants. And really, we want to see what is the impact of returning that score, as well as the digital technology, because we have all this done through Athabasca University. And does it prompt people to complete this online interactive learning module? So part of it with, like I said, Dr. Lindsay Carlson, who really has a lot of content expertise in this area, and they worked with a graphic designer to develop a little 30-minute online interactive learning module about genomics. So once you receive that score, it will be released to you and you will be invited to complete an online interactive module. So we're going to do some statistical tests on this. We're recruiting registered nurses, nurse practitioners, licensed practical nurses through all regulatory bodies across Canada. And we're very excited when we can get that up and running. So this is what I think I have another one with the QR codes that's there. If anybody wanted to take a picture of it, it's just a final few more tweaks, but you will see this in the coming weeks. And if you are interested, we really do hope that you will consider participating, because this is, going to be some really important and sort of really the first time that we've collected this type of data, and is all part of moving this knowledge form forward. Yeah, so I think I have in the next slide as well. Right, so if anybody is by their computer, that is the QR code that will link to it, and it'll take you right on that. And, once it's open, we'll have it open for probably about a period of 6 weeks. Yeah. So just talking about our next steps, this is really we really are just starting. Hopefully, like I said, we need infrastructure, as April mentioned about the policy catalyst grant. We're hoping to create some of these resources that are going to help guide nurses in practice, because right now that just doesn't exist. So we need more and more nurses to take on leadership roles, to engage in this, both, like you said, at all domains of practice: education, research, administration. And there are nurses in the US who are doing fantastic studies that using omics, and it's just they're true nurse research scientists. So we want to promote a ecosystem and a country, because things are happening in Canada as well generally, but it doesn't seem like nursing is moving with it to the extent that we'd like. But we're going to change that narrative, obviously. But the Center for Genomics and Policy at McGill University, Genome Canada, we have a lot of things in our favor. We just need to make sure that nurses have a seat at the table. And we should, because we know that central to nursing practice is person-centered care, and we have a role in this space, because there are very many nuances, as April can tell you, all over, because that was really the psychosocial processes is what she examined in her dissertation, and how people have very nuanced responses to this, and it's not a one-size-fits-all. Ultimately, once we get there, we want to have some intervention. So if we use our analogy of DNA, we want to create, right now that we have the priorities, that we say those are our RNA. So we're in the translation stage, and we want to yield lasting proteins, which is interventions that nurses can use in their practice. So that's really end of the formal part that we had today. We're going to open it up for any questions or ideas right now. We'd like you to reflect on some of these questions, and if you have any ideas, we'd certainly love to hear them. And just, of course, our acknowledgements. This is a group effort, and it's been the result of a lot of leadership, and we'd have to certainly acknowledge all those people who's been a role in this development.